Hey everybody, Mr. Murphy here, and I'm going to take a quick look at chapter 2.3, which is carbohydrates. Right off the bat, this little formula here is explaining the relative amount of carbons, hydrogens, and oxygens inside of a carbohydrate, and it's doing so in a way that highlights the fact that it's essentially water molecules trapped within these. But basically what it comes down to is the fact that there are a certain number of carbons and oxygens, and there are twice as many hydrogens in a sugar molecule. Think about glucose being C6H12O6, which I think is something that you should know, a valuable thing. And it basically comes down to the fact that whatever bonds the carbon's forming, remember it can form 4 bonds each and will in a stable molecule. So whatever bonds there are between carbons and oxygens or carbons and other carbons, basically all of the other open bonding sites are going to be filled with hydrogens, which is why we see more of those. We associate carbohydrates with storage and transfer of energy. This second point, transport stored energy within complex organisms, is important to understand, especially as we get into our future units in thinking about why we use carbohydrates and glucose and glycogen and things like that within our bodies, rather than just having a stockpile of, let's say, ATP. And essentially it comes down to the chemistry of it that carbohydrates are very stable molecules. Absent any sort of enzymes to facilitate their breakdown, they're a pretty good way to store up large molecules in a very stable and reliable way, unlike ATP, which is freely and readily giving away essentially the stored energy by chemically changing things around it. So we couldn't just have a stockpile of ATP, whereas we can have a ton of glycogen stored in our liver, and it becomes this really effective way to sort of store energy, much in the same way that like a potato is storing starch for a potato plant. So I'm saying you are a potato, essentially. There are other uses for carbohydrates. They are structurally important in the cell membrane and a variety of other places in and outside of the cell. And they're also important for signaling of identifying cells so that things like our immune system and other aspects of cells within our body can identify what they're actually bumping into based upon the characteristics of carbohydrates and other molecules on the surface. So at the very beginning, we have monosaccharides, which is the monomer of carbohydrates. There are 2 main types, pentoses and hexoses. Primarily, we've talked about hexoses. These are not terms that I would say to be terribly concerned about. Just understand that there are some differences in the structural makeup of different carbohydrates. So glucose, fructose, galactose, mannose, these are all different hexoses, C6H12O6. But we will also talk about ribose and deoxyribose being sort of those main components of RNA and DNA. So that's something just to be aware of. It's, again, not the main point of this unit or chapter. So here you can see the structure of our 5 carbon sugars, and here's the structure of our 6 carbon sugars. And even though all 4 of these here have the same formula, each one has a different shape to it, a different sort of where the extra carbons are hanging off here. You can see a fundamentally different structure between like glucose and fructose, and it's just the arrangement of where the directionality of these different hydroxyl groups and things are. It seems subtle, it seems like it shouldn't be important, but as we continue to grow our understanding of how the 3-dimensional shape of molecules really does dictate what's going on inside of a body or inside of any living thing, it should make sense to us that I don't need you to like memorize the structure of these relative to each other, but just to understand that by having these bonds occurring at different directions, that does make a different shaped molecule. And so when we think about enzymes in the next chapter, you think about how they facilitate, the bonding at the active site or the attraction at the active site with different molecules. Glucose is going to fit into a molecule different than galactose or mannose, which is a pretty important concept to understand, and just simply comes down to the fact that chemically they are arranged differently. So when 2 monomers of carbohydrates, monosaccharides, come together, that is a bond that is called a glycosidic linkage. This is the same as what we call peptide bonds in amino acids. It's just a term that we're a little bit less familiar with, most likely. It is exactly the same idea, though, of taking 2 monomers, we perform dehydration synthesis, and now we call this a glycosidic linkage. It's between 2 hydroxyl groups on either monosaccharide, we remove an OH from one side and H from the other. The carbon on this side here is going to want another bond. The oxygen on this side wants another bond, and so they will come together. This is a reaction that can happen spontaneously given enough energy and the right sort of circumstance of these monomers being in the right place at the right time. But in living organisms, it's predominantly happening with the facilitation of enzymes that are going to help to line these things up in a more favorable situation so that we don't need quite as much free energy in order to have the right sort of kinetic smashing together to make this happen. Because remember, a glucose molecule, it's perfectly happy in terms of the number of electrons in its different orbitals, and therefore it's not likely to want to rip off this OH from this carbon because there's a covalent bond. They're perfectly happy. They sort of have what they need to be stable. But when two molecules smash into each other, there's energy exchange, there's a potential for sort of the liberation of atoms, and so it can happen. Enzymes just make that happen a little bit more easily. More on that in the next chapter. So anyway, when we take two monosaccharides together, we call it a disaccharide. When we get several monosaccharides together, we can use the term oligosaccharides. These are often bonded to like proteins and lipids, and this could be glycoproteins or glycolipids. Not something that we're going to dive deep into with this unit or even really the course. We'll see examples of these. So it's a okay term to just sort of be aware of in the background, but not one that I need you to be able to differentiate. I'm not going to give you a molecule and like rely upon you to be like, oh, that's an oligosaccharide, not a polysaccharide or whatever. Polysaccharides are important, though, for us to understand that these are much larger carbohydrate molecules, and the three primary ones that we focus on are starches, glycogen, and cellulose. Starches being that energy storage molecule for plants. Think potatoes, think corn, like the actual inside, like component inside of a kernel of corn, and it's energy storage. You think about, why does a potato plant make a potato? It's a good question, and really it's energy storage. That potato plant is working hard for its ability to store up that energy so that the potato plant itself will be able to survive the winter until you, terrible monster, come and rip it out of the ground and turn it into potato chips or something. That's an energy storage molecule. Glycogen is our version of that in larger animals or really just animal cells in general. Glycogen gets used as the storage molecule for mammals here. So in our liver, we store up tons and tons of glycogen. It's much more branched, as we'll see in a diagram, than starch, which allows for more readily available energy. Cellulose is, as it says, the most abundant carbon-containing biological compound on Earth. It is the sort of structural backbone of plants. As it forms these long chains. So here you can see cellulose is big long chains of glucoses together. Meanwhile, starch and glycogen are these sort of branched chains. And here you can see a good representation of how that linear structure really leads to the specific function of cellulose as this tightly stacked and structural component. Meanwhile, the branches of starch and the highly branched glycogen are significant because the more branch points means the more end points. And the vast majority of enzymes that are facilitating the breakdown of carbohydrates inside of living organisms are working on those ends and have a shape that's so specific that it can't just jump into the middle of a large carbohydrate molecule and start breaking things down. It has to work from the end, one monomer at a time. So if we were comparing starch and glycogen, glycogen has so many more branches and is therefore found in organisms that are going to need larger amounts of energy more quickly, which should make sense, right? That's mammals and us needing to be able to, like, at a moment's notice, jump up and run away, versus a tree, which has some serious energy needs, particularly larger plant life, but not nearly the same sort of active need for energy that we have. Okay, here's some pictures of polysaccharides inside of cells. Enjoy. And that's the end of the section. So next up we'll have lipids, and if you have questions, let me know.